So we're going to talk about Chapter 1, Biology, and biology is the study of life. And we're going to go through what you have in your notes that pertains to Chapter 1 so that this can help you with your test that you have this week. So for Chapter 1, Biology is the study of life, and nature is everything in the universe except what humans make. And then you have life's organization, and how they organize life is they organize it atoms, then molecules, then organelles, and then cells, and then tissues, and then organs and organ systems. And then you have organism, population, community, ecosystem, biosphere. So let's go through each of those one at a time. You have atoms, which are fundamental building blocks of all substances. And you do have quarks, which are smaller than atoms, but usually in biology they don't talk about quarks. That's more like physics, but just realize that quarks are smaller than atoms. And so after atoms, you put a bunch of atoms together, you would get molecules, which are two or more atoms put together. That would be like a water molecule. Then you have organelles, and those are baby organs in a cell that perform specific functions similar to organs like in your organ systems in your body. And in a cell, you have things like the nucleus, the Golgi apparatus, the smooth endoplasmic reticulum, the rough endoplasmic reticulum, ribosomes, etc. And then after that, you would have tissues, and tissues are groups of cells put together. So in your body, your skin is considered to be keratinized, stratified, squamous epithelial tissue. So your skin is epithelial tissue, and that's an example of what a tissue would be. And then if you put tissues together, you would get an organ. And in your body, you have organs like your kidneys, and your lungs, and your liver, and your spleen, and your gallbladder. And then when those organs work together, like skin, hair, and nails, make up your integumentary system. And so that creates organs together, create organ systems. And then if you put organ systems together that function, then you get an organism, which is an individual consisting of more than one cell, one or more cells, because you can have single-celled organisms. But in humans, we're multicellular, and then you have things like certain types of protists like Euglena, amoeba, paramecium that are going to be single-celled. So an organism is anything that could be something that has one cell, which is single-celled, or could be multi-celled like humans, bats, etc. So then after organism, you would have populations, like populations of people or populations of penguins. So you would have the population, like all of the people in Wythe County, Virginia, would be the population. So that's the same species in the same place. And then if you had like a zoo, think of a zoo. If you had a zoo and you had all of the elephants and the tigers and the chimpanzees, that would be more like a community. That's the same place, but all different types of species there in the same, occupying the same space. And then if you get a community, whenever you group together communities, you get things like a lake or a pond or a forest, and that's an ecosystem. And an ecosystem is a community and its environment. So in an ecosystem, you have things like the pond has the non-living factors like the water and the salinity of the water and the pH of the water and the sunlight, and those are all non-living. But within the ecosystem, you have the living factors like the fish and the snails and those type of factors. And all of those factors interacting create that ecosystem. And then if you put a bunch of ecosystems together and you're talking about the entire Earth and all of its regions and layers and ecosystems and communities, etc., then that's going to be a biosphere. Used to, they didn't include organelles, but now they include organelles. So let's go back through the whole list from beginning to end. You would have atom, molecules, organelle, cell, tissue, organ system, organism, population, community, ecosystem, biosphere. And that's your order of the organization of life. So you have an overview of life's unity, such as all living things have similar characteristics. You have every organism in the world needs some type of input of energy, and they have cycling of materials. So you have food that goes into your body and you have endergonic reactions, but that food is going to give you energy, but then you're going to have to use that energy, and that's going to be an exergonic reaction. So every living being has to somehow get energy in some form or another, and then they have to utilize that energy. So you're going to have energy inputs and energy outputs. So organisms also sense and respond to changes and to their environment. So you have stimulus and response. Every organism is capable of sensing stimuli and then responding to that stimuli in a way that allows them to be able to survive in their environment. And the way that they respond and the way that they react is going to depend on where they live and changes throughout the year, but it also could be affected by the fact that maybe you change an organism from its natural environment to like captivity, like a zoo, for instance. All organisms also have DNA or RNA. Some have both. Humans have both DNA and RNA, but things like viruses are either DNA or RNA. So you either have an RNA virus or DNA virus, but they're usually not both. But all organisms have some type of genetic material, whether it be DNA or RNA, and we happen to have, as humans, both of those. So all organisms have DNA. The DNA that organisms have, and you'll learn this more when we get to genetics, all organisms have A's, T's, C's, and G's. So they all have the adenine, the thymine, the guanine, the cytosine. It's just that they have different numbers of chromosomes and they have those letters, those nitrogen bases in a different order. But every living being that has DNA has the same nitrogen bases that make up their DNA. So the essential basic structure is going to be the same. So all organisms have to have that energy. They all have energy inputs and outputs. They all sense stimuli and respond. They all have some type of genetic material. So they have to have that genetic material and they have to have that nutrition to be able to live and to be able to respond and to be able to regrow cells and to be able to rehabilitate themselves or regenerate new cells if they get hurt or injured. Like you do when you cut your skin, you're going to regenerate growth of new cells. And to be able to do that, you have to be able to have the ability for your cells to be copied, which means you have to have some type of DNA that allows itself to be copied. Whenever you're looking at organisms, you're going to have organisms that are producers or organisms that are consumers. So producers are going to be like plants, and they're also called not only producers, but the other name for them are autotrophs. So they automatically produce their own food via sunlight. And there's some organisms that will be chemoautotrophs that will use chemicals such as methane in order to maintain and make energy for themselves and be able to sustain life. But plants are going to use the sunlight, so they're producers or autotrophs. Then you have humans that have to go out and get energy, and they're going to be heterotrophs or consumers. Those are the two names for something that has to go out and get its food and something that can't just lay out there in the sunshine and make pizzas and hamburgers. So you have to have the ability to go out and get food and maintain it and consume it. So those are the basic overview of what's in your notes on chapter 1 in regards to organizing life and biology and how life is organized and the basic things that organisms are going to need on the earth. So let's turn over and talk about scientific investigation or scientific inquiry, or what you've probably heard it called scientific method. Whenever you're doing an experiment, you're going to have to set up certain protocol to be able to investigate. And you're going to try to investigate in a manner that makes errors minimal. And you're always probably going to have errors that you're unaware of. And we've discussed this in class, but you're going to try to minimize those as much as you can. So when you have an experimental setup, you're going to have a variable, which is going to be the single characteristic in a set of individuals that differs from the control group. So you're trying to test just like you did in class with the earthworms or the mealworms or the waxworms or the redworms. You're testing different dry and liquid ingredients, which is what you did in class. Those were your variables. When you tested an earthworm with hot sauce versus oatmeal, those were your different variables that you tested. Your control is going to be your standard for comparison. So whenever you observe the earthworm or the waxworm, the mealworm or the redworm, when you observe them and they were just in the dirt, when they were just in the oatmeal, when they were just in their natural environment, those observations you made without having them exposed to something was your control. And of course, we talked in class about constants. Whenever you were testing these organisms, you wouldn't have wanted to have tested one earthworm in really cold temperatures and one in really hot temperatures. You tested them in generally the same environment within the same classroom. Even though there can be little microclimates or an air vent right over the earthworms, you generally have the same environment, which is your constants. So your experimental group was the group with a single variable that you were testing. So those variables that you used were the different substances that you exposed your organisms to. So now You had your control group, which is just you observing the organisms in their natural environment. So control versus variable. The variable was what you were changing. The control was what you were keeping the same, your standard for comparison. And then your constants were all the things you kept the same, like the same temperature, the same testing protocol. If we were doing a very specific experiment, you would have used the same amount or quantity of the liquid and the dry substances that you exposed the earthworms or the other critters to. You also are going to have different things that you're going to look at as we go throughout the year. And sometimes, most of the time, you're just going to have one variable, but there are going to be a couple labs that we're going to have more than one variable. So one of the things we need to talk about is the different types of variables. You have independent variables and you have dependent variables. And sometimes people get these confused, but what you need to think about is this. Your independent variable were like the things that you exposed your earthworms, mealworms, waxworms, and redworms to. So the different substances. That was your independent variable. Different things you exposed them to. Your dependent variable were the different results you got. For instance, think back. All of your critters did not react the same to all of the ingredients. They all reacted differently. So those different results and different reactions you got, those were your dependent variables. So independent is what you change. Dependent is the different amounts you get or different responses you get at the end. And it can be wholesale different. So for instance, if you had Miracle-Gro and you put different amounts of Miracle-Gro on 10 tomato plants and on one you just put water. Your independent variable would be the different amounts of Miracle-Gro you put on each plant. And then how would you measure how well each one grew? Well, you're going to take a measurement tool such as a meter stick and you're going to measure how tall each one was. And you're going to measure them the same time, each day, the same way. So your independent variable was the different amounts of Miracle-Gro that you put on each plant. And the dependent variable was going to be that all those plants aren't going to grow the same size. So your dependent variable is going to be how tall each one grew. And you're going to have different heights for each one of those. So that's your dependent variable. There's different examples that are in your notes that you can read about. There's peacock butterfly defense mechanisms against predatory birds. And there's some different examples on there that they discuss in your note packet and within your PowerPoints, but I'm not going to discuss those here. When we were in class, we talked about sampling errors and we talked about different reasons why you might have errors. Whenever you're trying to do an experiment, like I said earlier, you're going to try to minimize the errors as best you can. So you're not going to want to intentionally do anything that would screw your results. And in class, it's not as big a deal, but if you were doing this and you were a research scientist, it would be a bigger deal. Okay, that's chapter 1, and that is just some overview of your notes. And then later on, I'll have an actual test review on this slide as well that will have some actual sample test questions for you.